Hi everyone, it is a pleasure for me to present at the OHBM 2023. My name is Eduardo Garza and I work at the Institute of Neurobiology, UNAM in Mexico. I have nothing to disclose. So very briefly, substance use disorders, as we know, are a worldwide problem, a neuropsychiatric disease with the inability to control the use of substances, which can go to moderate to severe that leads to an addiction, and the treatment is very limited. And cocaine, which is the one I'm going to talk about, is a stimulant drug. There are plenty of limitations in substance use disorder research, especially in human studies and using neuroimaging, but also in all neuroimaging research. This recent study by Rasgado-Toledo et al. In Nature Neuroscience show that a lot of the neuroimaging data sets out there and studies are done in white population, which is fine if you're interested in that. But in any case, the United States of America and the United Kingdom, as the associated data sets, are widely used to make inferences in a global context, which is completely biased because there is more people in the world and there are more types of people and they are different. And of course, in substance use disorders, these populations are very different. Every country has a different socioeconomic background, education, stigma, and the substances they use are different. So everything should be, in a sense, balanced. And right now, it is biased.
And as a Mexican, of course, I'm very interested in the Mexican population, especially because it is very different from the US or the UK population. So there have been studies in Mexico about, for example, skin color as a phenotype, because we don't measure race in this case. And it surely shows that to the left, for example, it shows that the skin color is perceived and also objectively very normalized across the whole country. And you can see here in this picture as well to the right that this is the most common type of Mexican phenotypes you will see across the country. There might be more, of course, more extremes, but in general, it's a very homogeneous population. This is important for research as well.
And in terms of substance use disorders, it's very different between countries. And this is an example. For example, prevalence between US and Mexico of consuming any illegal drugs in the US is 50%, across a lifetime. And in Mexico, it's only 10%, for example. But there are more differences. Like in the US, there are more like alcohol, cannabis, and prescription painkillers are the most common drugs. And in Mexico, it's actually very similar, except there are no prescription painkillers. So we don't have that issue. We have other issues. So that will be very different. And the results you get are very different in a lot of things. For example, initial age of consumption is lower in the US than in Mexico, and so forth. So in general, we can agree that we need to study different populations so we can actually know what's going on in terms of brain pathology and what treatments can work for different populations as well. And especially when compared to underfunded and technologically underdeveloped countries such as Mexico.
So with this in mind, I wanted to introduce the SUDMEX CONN, a data set of substance use disorder patients with clinical, cognitive, and MRI measurements in Mexico.
So I came to work to the National Institute of Psychiatry, and this study was conceived by my colleague Jorge González Olvera, psychiatrist and a researcher in Mexico City. I became the co-PI of the study when I got there, but I wanted to do some changes. So the National Institute of Psychiatry is a great place to work. It's a public center that serves the whole country and combines clinical attention, clinical and basic research with social studies and education, and they have a scanner. So the original project was just funded as an MRI dataset of many substances use disorder. So when I got involved, I became the co-PI of the project. And the first thing I thought was to make an open clinical dataset. I didn't know at the time how rare an open clinical dataset with MRI was. I was just starting to get into the whole open science thing, and I thought it was great. But when I decided to make it open, it meant that everything had to be done, properly with attention to detail, which proven to be very difficult, especially underfunded. But I think it worked. I wanted the dataset with clear and complete clinical data as much as possible and high quality MRI to go along with it. I thought I was doing the norm, yet found most clinical datasets are incomplete and MRI sequences are not the best. Also, it seems most researchers, either due to laws or other constraints, can't release raw data. But this is something I wanted to do from the start. So we basically released this a long time ago, but right now we had a publication last year, in scientific data, which describes the data set. You can find it in scientific data. And we have high quality clinical, psychological, and MRI data from cocaine use disorder patients and controls. So just briefly, in terms of the clinical data, we have general information, we have psychiatry information, very thorough, done by psychiatrists and psychologists. We have addiction and data, and at the moment we have a sample size of 64 controls and 74 patients. In terms of the cognitive data, we have everything done in a computer, and we have working memory, cognitive flexibility, inhibition, decision making, and theory of mind data that you can use right now. So the data was acquired using a 3 Tesla Philips Ingenia scanner with a head coil of 32 channels. In terms of T1 is very standard, the fMRI, the resting state fMRI as well, with a TR of 2. However, we do have a Hardy multi-shell diffusion acquisition with a B1 of 1000 and a B of 3000, which it's very different from other data sets. And I think it's an advantage, so we can use like Naughty, for example, to study microstructure. So I basically browsed a little bit about the data set, but I'm mostly interested in telling you about the challenges and other things and how open science helped with this. So in terms of the infrastructure, we did have OMRI, which was great. However, there was a lot of problems. For example, although I have access to the Philips scanner, it was just 99% of the time was used for clinical studies, and there was no dedicated time for research. So I was charged by the hour, and there was no technician or physicist who was an expert on research sequences who could help me, and there were no research sequences. So I had to create them myself with the help of other colleagues that have more expertise than me, and in some of them. Like for example, the diffusion was done with my colleague Luis Concha. The time I used to create the sequences was in between other clinical studies because I had to be in the scanner while they were preparing the patient, or at other times when they were not scanning. However, they weren't very happy to see me all the time, but I had to be there, so no problem. The only good thing was of course, the price is very low in Mexico compared to the U.S. I've heard, so that was a good thing at least. So piloting in research, human research, is very important. However, I was given the chance to pilot one healthy participant, and with that we had to start acquiring patient data. This is the reason why our first T1 weighted images had a different voxel size, it was lower, and why the diffusion wasn't acquired until the sequences were ready. So not everyone has a diffusion because we started later, but I couldn't get time to pilot because I had to pay for the time and I didn't have that much funding. So it was a problem. They weren't used to being there. And I think a lot of the other people in the developing countries can relate to that. So because we use clinical MRIs, that's the problem. So the recruiting was very challenging, as in Mexico it's not possible to pay volunteers, or it's very difficult to pay them due to administrative problems. So basically we had to print ads and send them all over Mexico City, which is great because it's a huge city. The controls were the hardest to get, I think, because we wanted to match age, sex, and education, and possibly socioeconomic level as much as possible. And it was hard. We were asking our everywhere. And the only payment that subjects had was they received their CD with the T1 weighted image, and of course a thank you from us. But most people were happy to, in Mexico, many people are happy to just contribute to research, which is great. So at the beginning, we were only 4 working on the project, but with time, we got some great students, a great master student called Tania Balducci, and my other great bachelor students from psychology that really help acquiring and getting this project going. In Mexico, master and PhD students are paid by the government, which is great. However, postdocs are very uncommon. So, and it's also difficult to get students as well. So it was great that I could get really great, brilliant bachelor students to work with this. Data management was done by myself as well as quality assurance. Finally, in terms of bureaucracy, so this is also something that a lot of people in Latin America will probably know, and also in other developing countries. So the system was not made to pay MRI upfront. Like I had to pay for each patient, so I couldn't use funds like right away. So we had to get creative with that. It was hell, I would say. to manage that. We had to manage our money and all that, so that's also very common. A subject had to be included as a hospital patient and thus make all the paperwork for a patient, even though there were no patients sometimes, like with the healthy controls, we had to do everything. So this type of institution was made for research, and yet they did not make it easy for researchers in human studies. So, and that's very common. I would say that's very, very common. So a lot of people will have this problem as well. And you have to sort it out, basically. So what about open science? So right now, research needs a lot of external validation, especially if we want to generalize everything we get. So in terms of substance use disorder research, we need to generalize everything so we can use, these results for treatment, management, etc. And right now, external validation is very low, especially in neuroimaging. Open science as a set of principles and practice that aim to make scientific research from all fields accessible to everyone for the benefits of scientists and society as a whole can accelerate discoveries. And I truly believe that. And that's why I wanted to do this open. So once we had some data, we published one paper using this part of the data. But once everything was finished, I realized, because usually you want to finish everything and then publish some papers and then, get the data open if you can. But I realized, I didn't have that much time. I didn't have that, I wasn't that smart, really, just to do everything that was to do with this data set. And I didn't want to, keep it for myself. So I decided that the best thing to do was to release everything as raw as possible. So I put everything in the bits framework that was starting at the moment. So I uploaded the raw bits and data, MRI data to OpenNeuro, and then the raw clinical and quantitative data to Zenodo. But it's not completely raw. We had some work with it so people can actually use it, and that is easy to start working with. So OpenNeuro and Zenodo have been my favorites because they're easy to use and they don't have many conditions. I was criticized by some colleagues for uploading raw data when I could still squeeze it for more papers, but no, I thought it was a waste of resources just to leave it there for me and my small group. And I also learned about Enigma Addiction Framework, which they have a lot of they share clinical data from many patients. And the only way to enter is with, sharing data. So that's how I did it. So it's great. It was a way to completely share the data with other people and it's, right now being used. And so then people starting, publishing data, publishing papers with this data. And there are about 7 published studies that I know of, and I think there will be more with the Enigma. And there are more in the works. People that were downloading the data and referring the first paper. So that's why we decided to send it to Scientific Data so people can actually know where to, what the whole data is about. And we quality assess the data for people as well. So, and I realized this is important things. Researchers that approach me by email asking questions about the data, which may realize mistakes I did, and correcting them. This was very valuable. Even if the data was free to use, still researchers told me that they were, they wanted to use the data, and if it was okay. I mean, it was okay, but everyone asked. A lot of them asked me before, so it was interesting. Some researchers also wanted me as a collaborator because it was easy for them to navigate the data, or because they did not have the clinical expertise, but they had the technical expertise, which was great. I mean, from then I have great collaborations, and so I think this has taught me a lot about open science and how I didn't have to actually keep the data for myself until I squeeze everything out. People were great. And right now, for me, the greatest thing is that right now I'm working with researchers from Iran, India, China, U.S., and Canada, to solve different questions that they thought about, which is great because I've never thought about this question. So it is an amazing collaboration, and I think it will only get better. So in terms of future directions, we already moved forward with another data set that stems from a clinical trial that we performed called the SUDMEX TMS. which is now under review but already accessible as a preprint with the same philosophy. It's an open arrow, you can download it. It has very similar sequences, easy to use and as raw as possible. I believe that substance use disorder research will greatly benefit from the inclusion of other populations, especially females. I think that's also overlooked. And by inclusion of other populations and females, we can find convergent and divergent phenomena in this, in addiction. I mean, as an idea, if developed countries wanted to help with this, you can also fund research in developing countries. We have the expertise to do research, we just sometimes lack the funding. So it will be a great help for everyone. I think it will be a win-win situation, and it will help improve our understanding and treatment of substance use disorder worldwide. So I just want to thank everyone who has worked with me throughout the years. This is part of my team at the moment. However, I want to thank everyone that has worked with this and that collaborated with this research. Thank you. And thank you for listening. Thanks again for listening. And here is a QR of our lab if you're interested in our research.